Hi everyone, it's Mr. Sinti, and today I have the pleasure to be discussing with you a little bit about pregnancy, and it's the very first part of pregnancy. If you consider pregnancy being 9 months, that's approximately correct. To be more accurate, it's about 9.5 months or 40 weeks. And so I want to talk to you a little bit about that and embryonic development and the first, basically, a discussion of the first trimester of your pregnancy. But the thing is, it's so exciting about having a baby. One of the first things that you want to know when you have discovered that you are pregnant is when is that baby coming out? And so the doctor will count 40 weeks. And so you're like, wait a minute, I thought it was 9 months, but it's really like 9.5. But it varies in individuals. And so basically what a doctor is going to do though is going to start counting from the first day of your last period. Okay, so this is what we know. And so why would you do that though? Like, why would you count like the first day of your last period? Well, usually that's known, and the mother will be able to speak to that. And so technically, as you may imagine, you don't become pregnant during the period. It really happens around ovulation time where you're conceiving. And so that's around the second week of a normal cycle. So when a doctor's telling you you're 4 weeks pregnant when in fact you've been pregnant for just 2 weeks. So that's that. So this embryonic stage of development, or the first trimester, is really kind of defined as really the time between uh, when you're fertilized and about 8 weeks. Okay, so then what would you call this after 8 weeks? 8 weeks is around 2 months. So after the second month of pregnancy, then you'd start talking about fetal stages or fetal development, and then obviously when the baby comes out, or if the fetus comes out, it's a baby or a newborn. And so we're talking about the first trimester, and this is what an embryo looks like in 5 weeks right here. So it's kind of awesome. See the head and the vertebrae are starting to form, and here's some internal organs. And so you can see here again the head and the body, and you can make out various organs right here, and some of the limbs are starting to form. And that's what's interesting is the feet and hands are basically solid, and then the cells sort of die off and separate the fingers and toes of the phalanges. And so during this first trimester, during embryonic development, all of the internal organs or major ones are gonna start to develop. And basically in the fetal stages, there's some cool stuff that happens, but basically it's just going to enlarge. And so what you can see here is that the embryo is floating in this membrane called the amnion, which forms an amniotic sac, and the baby is floating, basically suspended in this amniotic fluid, which is cushioning and protecting the embryo during its development. And what you're going to see notice here is the umbilical cord, which is going to be connecting to the placenta, which is a really cool organ, which is a combination of blood vessels from the embryo and from the mother, and that's where gas exchange takes place. And so this particular video will not be a discussion of the placenta, but I will certainly attempt to make one specifically on the placenta because it's just that good. And so what I wanted to start off was sort of a transition. If you've been following a previous video on fertilization and the cleavage period, what's happening is this zygote is basically cleaving into this first couple of cells called the morula, and then it's called the blastula, and it forms this fluid-filled structure called the blastocyst. And it's basically the blastocyst which is going to implant in the endometrium, which is the inner uterine lining of the uterus. And so these outer cells of the blastocyst are referred to as trophoblast cells. Yeah, and it's these trophoblast cells which are some of the ones that are first to differentiate early in pregnancy and eventually become a membrane called the chorion, which will eventually differentiate into the fetal portion of the placenta. And so I know there's a lot of terms involved in embryonic development. Don't be discouraged, only be encouraged, because basically what the terms are trying to do, sorry for preaching, but what the terms are trying to do is clarify our understanding. And it's not to intimidate, or it's basically just to be able to really focus on what's happening specifically. And so there are many new terms that will be introduced. So the trophoblast cells are these outer cells of the blastocyst, as I mentioned, and this is actually showing the blastocyst implanting. This is inside the uterus. This is scanning electron micrograph showing how the blastocyst will begin to implant inside the uterus. And so basically here we have it. And so here's the, when we talk about the outer cells, we talk about these trophoblast cells, okay? And then there's this fluid filled inside. And these cells in here, these green ones, are referred to as the inner cell mass, or embryoblast cells. Now these are very important because it's these inner cell mass of cells, or ICM, which are basically going to become the embryo. So this develops into the actual embryo right here. And so those are particularly important. Again, a cool micrograph of that. These are the outer trophoblast cells, and these are the inner cell mass cells, which will eventually become the embryo. So these outer ones are the trophoblast cells. So soon after fertilization, the embryo develops into this hollow ball of cells called the blastocyst, and then that's what's implanting into the uterus. But it's these inner cell mass cells that are really cool. Just a little side journey on these inner cell mass cells. You might have known of them before. These are quite famous. The ICM cells are sometimes known as embryonic stem cells. And they're stem because they can, like a tree stem, branch off into and differentiate into all of the different cell types of the embryo and ultimately into us. So they can, at first, they're all the same, but then they can develop into neurons or adipose tissue or muscle cells or white blood cells. So they're really, really important because, as it turns out, in the late 1990s, researchers were able to extract these ICM cells, or embryonic cells, and basically culture them in petri dishes. And we found out that they grow best on fibroblast cells, which are a type of connective tissue. And so we can actually grow these. And so we can grow embryonic stem cells. And why would we do that? Because they're pluripotent, as I was mentioning before. They can develop and differentiate into other cell types in the body. And if we're able to manipulate these stem cells and grow them, because of the fact that they're very plastic, meaning that they can actually develop into all cell types, even more so than our adult stem cells, which you might be familiar with, which are found in the marrow of our bone, which can differentiate into different blood types. These particular embryonic ones are really important because understanding how they do that, it's not clear exactly how they're able to differentiate like that, or what are the mechanisms that cause it. And so it's one of those areas where there's a high degree of research. And so there's a lot of unsolved problems, which makes it really interesting. And so some of the basic research is trying to figure out how this works. And there's an urgency because you might know this. This is kind of obvious that there's implication and importance for human health because, the thing is, we have damage to our body. We can be, even if we're healthy now, we could be in a car accident. Something can happen. We can have a heart attack, which would destroy some of the muscle cells in our heart. We could have a stroke and cause some neuro damage in the brain. And so, as it turns out, if we use these stem cells, we might be able to insert them into damaged tissue, and they can regenerate and become and replace heart tissue, and maybe cure some damaged tissue in the future. So there's a lot of hope there, embryonic cells. So getting back to the embryology, one of the major events that happens to the blastocyst is that there's an invagination. There's like a folding inward of the embryo, and this inner cell mass in here, which is eventually going to become the baby, forms this embryonic disc, and there's three distinctive layers that form in this embryonic disc, and so this structure is known as the gastrula. So again, sorry about all the terms. So what happens is the blastula forms the gastrula, and then this embryonic disc, which forms these three primary germ layers, which are going to become all of the organs of the embryo. And so just a little discussion of this. And so here's the blastocyst with the inner cell mass, and then there's this invagination forming the embryonic disc, which is the gastrula. And so these primary germ layers will then form all of the organs of the body. And so they're called, the outer one is called ectoderm, and the middle is mesoderm, and the inner cell mass is called endoderm. So it's ecto, meso, and endo. And so you can really get into this if you're studying embryonic development, but I'm just basically going to give you just an outline of this. So the ectoderm can eventually form the cells of our epidermis, of our skin. It can form the neurons of our brain. Some of these the germ cells over here on the side can form sperm and egg. The endoderm can form portions of our lung, some glands in the body like the thyroid, pancreas, and the meso in the middle can form muscle cells, eventually some portions of the kidney, red blood cells, and again, part of our digestive tract as well. And so just to sort of continue this discussion of this, at first, again, sidebar, at first it becomes like, well, do I need to memorize or should I need to know this? Depends on what you want to remember. Personally, in my estimation, I don't know that you do need to recall this kind of what the ectoderm becomes, because simply you could look it up. And so I just bring it to your attention simply because of awareness, but don't feel obligated to recall all things. I don't think that's the correct pedagogical approach to this. I think when you can look something up, if you're overwhelmed, like I can't remember that the ectoderm produces portions of the nervous system, part of the sensory organs, or line the mouth and anal cavity. I can't remember that. You can look it up. Also, the mesoderm mesoderm becomes muscle, as I mentioned before, some bone. Blood comes from the mesoderm, kidney, and then endoderm produces the digestive tract, respiratory tract. So this is what we're getting at. And so what's happening, big picture, is here we are in the uterus, and so we're implanting. And so this the gastrula is forming here. We have the embryonic disc right here, which is made up of those three germ layers. And then eventually what's going to happen is the trophoblast cells will eventually become something known as the chorionic membrane. I'll explain that as much as I can. The chorion will then evolve into villi, which are extensions into the endometrium, which will eventually unite with blood vessels from the mother to become the placenta. And I know that's somewhat ambiguous, and so I'll try to make a separate video on the placenta because it's just an incredible thing. But I also want to talk to you a little bit about, you might be familiar with the fact that there's an umbilical cord which connects the embryo to the placenta, the forming placenta, which basically has blood vessels. It has two arteries and a vein, and that's going to help provide oxygen and nutrients and bring back carbon dioxide into the mother. And so everything that the mother is eating and breathing is going into the baby via the umbilical cord. Now, you might be surprised, but we have a couple of other membranes. I mentioned this amniotic membrane, or amnion, which forms the amniotic sac, and this blue is the amniotic fluid. But there's also a couple of other membranes. There's the yolk sac and something called the allantois that I want to talk about. And so here's another picture showing the embryo in here and the various germ layers over here. And then it shows the chorion forming, and eventually it will become the placenta. And here's the amnion, which will then eventually surround the embryo and cushion it with fluid. Okay, and so close-up of what we were talking about before. And so I'm going to run through some of these membranes. During this time, this membrane that I want to focus in on, this amnion, is particularly important because this amniotic membrane, most people know the amnion because they're familiar with the amniotic sac in this fluid because, again, during delivery, one of the very first stages of delivering a baby is that, you may have heard of this, is that the bag of waters break. And so that's the amnion membrane rupturing, and the amniotic fluid is leaking out the birth canal. It's one of the first signs of labor. But not only that, the bag of waters, but it surrounds and cushions the embryo, as you can see here, and it's a protective environment. One of the other reasons that I think most people know about the amniotic sac is that there's a procedure known as amniocentesis. Now, amniocentesis is sometimes referred to as amniotic fluid test, or AFT. It's a procedure in which the physician will use a syringe with a needle and actually go through the skin, go through the muscular wall of the uterus, and into the amniotic sac. And so some of the amniotic fluid is drawn into this syringe. And so why would you do this? Well, so the fluid, the amnion, contains some of the fetal cells that have sloughed off. And so there's some fetal cells that are floating around in the amniotic fluid. Now, why would you do this? Because you could actually then take a look at that amniotic fluid and look for some sort of problems that might be occurring in the fetus. Like, for example, you can examine it for genetic abnormalities, like an abnormal chromosome number. And so to assure that there's no damage being done to the fetus when a needle is entering into the uterus. I mean, this is kind of a blind procedure if you think about it. You're shoving this pretty long needle into a thick uterus. You don't want to actually penetrate the fetus. And so this is accompanied by ultrasound. And so when you have an ultrasound image of what's going on, you could be more assured that you're drawing out just the amniotic fluid and some of the fetal cells. And again, you take those cells and then spin them down, and then you can culture them and look at chromosomes. And so just want to take a second and take a look at what I'm talking about, just to make sure that everyone's clear on this. This is a typical ultrasound. So while the ultrasound is being performed, this is when the physician's able to insert the needle into the body through the uterine wall and draw out some of this amniotic fluid, which is where I'm pointing right here. So all this dark area is the amniotic fluid, which is surrounding the baby, just to make sure that you're not gonna hurt the baby in any way. And so let's take a look here. Again, what I was mentioning is that when you draw the cells out, you could then take a look at those cells and find some that are mitotic. In other words, there's chromosomes being formed. You can take a picture of those chromosomes, which is called a karyotype, and then you can align those homologous chromosomes together, and you could actually, if you will, if you know your genetics, you'll know that if there's two very large chromosomes, 45 and 46, you're going to have a girl. So you can determine sex by doing an amniocentesis. But really, what you want to do is look for chromosomal abnormalities to make sure that the baby actually has 46 chromosomes and not like 47. Like an example of that would be if you count them up. Look at this baby girl, because she doesn't have a Y chromosome, 2 X's. This baby girl has 47 chromosomes, and she has 3 of the 21st pair. And you're supposed to have a mother, father, mother, father. In this case, there's 3. And so this particular genetic syndrome is known as Down syndrome. And it's one of the most common genetic disorders. And one of the reasons that it's common is that get back to this, is that these particular chromosomes are very small. And it's sort of like socks in a dryer. They're the ones that always get lost. And when I meant get lost, I mean during um, egg formation and sperm formation, one of the processes to create those reproductive gametes is meiosis, and so there needs to be, the chromosomes need to separate during anaphase 1 of meiosis, and so sometimes that doesn't happen properly, and there's a non-disjunctional event resulting in an abnormal chromosome number in the gametes, or aneuploidy, and so this particular abnormal chromosome number is one extra one, or trisomy. You're supposed to be diploid, or 2. So this is trisomy 21, which is Down syndrome. And so when do you perform amniocentesis? It's usually done between the 16th and 22nd week of pregnancy. And so, this is past the embryonic stage. So this is like fetal stages we're talking about now. But women will be encouraged by their gynecologist and obstetric physician, or OBGYN, if they pose a particular risk. In other words, if the female, the mother, is of a higher age, because as it turns out, there's a correlation between the mother's age and the incidence of Down syndrome. And so again, this is a good picture showing here. Here's the bladder. Here's the uterus. Here's the placenta over here, umbilical cord. So the idea is to draw out some of the amniotic fluid using ultrasound to make sure that you don't hurt the baby. Here's the cervix right here, and then the birth canal. And so I thought I'd throw out a few statistics on this and of Down syndrome. As you can see here, there's a real correlation in mother's age when mother gets up a little bit older. Not to say that you're old when you're 45 years old, but you're old in terms of when you'd want to start your family. And so the incidence of Down syndrome is a little high at that age. And so doctor's going to want you to, or suggest that you get an amniocentesis if you're in this particular area, to make sure everything's going well with the baby. And so I mentioned a couple of these other membranes in this chorion. The chorion starts, this membrane right over here, starts to develop villi, or extensions, finger-like extensions. And ultimately what this membrane is going to be doing is helping exchange oxygen and carbon dioxide with the mother and the baby. And that's going to eventually form into the placental villi. And so here's something that's kind of interesting. I mentioned this before, that there's another membrane called the yolk sac. Now you might be thinking, a yolk? I thought only eggs of a chicken had a yolk, but no, we have one as well. It doesn't remain all during the development of the fetus. It happens early on, and it's shown here. And what it does is that it provides a nutrient-rich supply of food for the embryo, but eventually it disintegrates because the food and nutrients are going to be provided by the placenta. And so let me come back here. And so another function of the yolk sac is that it's a primary site of red blood cell production, and that is, you might imagine that because normally in an adult, red blood cells are produced in the bone marrow, and so obviously there's no bone in the embryo, and so red blood cells are produced there. So here's a cool picture of the embryo. Here's the placenta, the umbilical cord. As you can see here, here's the amniotic sac and the amniotic fluid. And so the yolk sac I was mentioning before forms very early on, and it's the site of red blood cell production. That's pretty important. And it also gives rise to some of the white blood cells as well. And so the allantos, another membrane, forms during the 3rd week, and that helps to join along with this connecting stalk that will eventually give rise to the umbilical cord, which is made up of the 2 arteries in a vein. And so a better picture of that. Here's the allantos right here. And so what does this do? It's basically sort of like a little bag that is the primary structure involved in nutrition and excretion early on, before the placenta is totally formed, and it's to collect waste from the embryo, and so it functions in those capacities. And so I think one of the things that is most interesting that people want to know about embryonic development is like, well, what's going on with the heart? When can I start to hear the heart? Well, the fourth week, pretty early, the heart begins beating. The fourth week of your pregnancy, and so the head, the jaws begin to form. As mentioned before in the previous picture, your limb buds start to form, and eventually this chorionic membrane becomes very villi-like, and it's going to eventually form into the placenta. And again, it's a separate video on that chorionic villi becoming the placenta. And so, actual size of the embryo is really tiny, like a grain of sand or a grain of rice. But ultimately, it's looking pretty advanced, if you ask me. And so, here you have it, embryonic stages. It's your embryo and your amniotic fluid right there, and your chorion and your allantos and your yolk sac. And then eventually, and I hope this gets your curiosity going, like check this out. This is going to be the placenta. And so we want to know all about how the mother's oxygen is going to come into the baby, and the baby's carbon dioxide is going to come out, and the mother's food's going to come in, and etc. So that's going to be a cool discussion of the placenta. So I hope you enjoyed this video on embryonic stages of development. Thanks for watching.